Hello, so this is the start of a series of live streams I'm going to be doing, going through all of the A-level biology AQA content. I'll be having sort of interaction, so you can message in the chat whenever and I'll respond as soon as I possibly can. These will be uploaded as videos, so you can go back through, watch them again, and catch anything you didn't miss, and feel free to then in the comments go through and ask questions. So to start off with, we have carbohydrates. You've got three main types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. There are four main types of monosaccharides: alpha and beta glucose. They're indicated by the symbols there and there. You got fructose and galactose are the other two. These will then combine to make three disaccharides, which we'll talk about later: maltose, lactose, and sucrose. And then glucose can be polarized into three polysaccharides: starch, glycogen, and cellulose. Again, we'll talk about them later. But going on to monosaccharides. So all of them will have your general formula of C6H12O6, so there. And then the only structure you have to know, so the drawn out structure, is of alpha and beta glucose. You can see them here. So that's your alpha glucose and that is your beta glucose. So the only big difference is that position of your OH. So on the alpha glucose, it's on the bottom, and on the beta glucose is on the top. This will come in when we talk about polysaccharides later. So you also have to see these numbers written around them. They are your labeling of your carbon. So each point on this hexagon is a carbon other than this oxygen, and then you've got this additional carbon up here to make your 6. So any questions about that? No. Disaccharides. Disaccharides are formed by condensation reaction. So this is your removal of H2O and forming a glycosidic bond. So this will happen between carbons 1 and 4 even. So carbon 1, carbon 4, 1, 4. And these will produce your disaccharides. Di meaning 2, so 2 sugars. And they're again broken apart by hydrolysis. So your addition of water to make your two molecules again. To break the glycosidic bond to make your molecules. So your molecular formula for these is 2 glucose added together minus water. That's C12H22O11. While both maltose and lactose are reducing sugars, so that means they can be oxidized, or they give off a hydrogen, I'm pretty sure. Sucrose cannot, and we'll talk about a bit about that later. So polysaccharides, there are three: starch, glycogen, cellulose. Starch and glycogen, they have these large molecules storing glucose. They're branched, got many ends, so they can be hydrolyzed quickly. They're osmotically inactive, so they won't affect the movement of water going into the cell. And yeah, they're broken down very quickly because they've got brand new branched ends. This is because they form not only 1,4 bonds, as shown on this bit here, they form 1,6 bonds as well. So they can be hydrolyzed very quickly to produce glucose, which can be used for respiration energy. Cellulose is slightly different. This uses beta glucose as opposed to alpha glucose. So this means we have to alternate each one. So look at this position of this O here. It's now on the bottom over here. So we can form this sort of shape. So these provides long continuous microfibrils, which are then used in the structure of the plant cell wall. They're held together by hydrogen bonds, and they are unbranched, so they don't form the 1,6 glycosidic bond, only the 1,4 glycosidic bond. Here is a little table talking about it. So the type of glucose used, so alpha, beta, alpha, which sort of bonds, spiraled, such when it forms the sort of 1,4 bonds, they slowly coil. Here, you don't have any. Where they're found, so starch and cellulose found in plants, glycogen is found in animals, and their job, so glucose storage is the main one for starch and glycogen, and cell walls is cell rigidity, so it doesn't burst and helps hold up the plant. So, how do we test for sugars? The main one is the reducing sugars test. So this is when you add a sample to water. So add your unknown sample to water and let it dissolve. Then add equal volume of Benedict solution. So this is a chemical that will react with the sugar. You'd heat it for 5 minutes. So put it into a water bath, probably like 40 degrees C. And then if the reducing sugar is present, you will find a brick red precipitate forming. So, I can't speak. So the precipitate form, this can be sort of measured quantitatively or qualitatively. So qualitatively, you just look at the colour. So the more colour, the stronger the colour, the more concentrated the sugar. Quantitatively, you can measure the mass of the precipitate formed to get an exact number. So, if we were to test the sucrose, this would turn up as blue because it's a non-reducing sugar. So now we have to look at tests for non-reducing sugars. So, first thing we would have to have done is test for a reducing sugar to make sure it's not that. So if we want to identify a solution having sucrose, we have to make sure that it's not a reducing sugar first. So we do that test. If that comes back negative, then we'll get a new sample, add some like hydrochloric acid, strong acid, and boil it in that acid for a certain time. Then neutralize it with a base. So any base will do, or alkali. And then you are going to add your Benedict solution again, heat it up in a water bath, and then you get the same results with the formation of your brick red precipitate. Cool. Starch test. So this is a specific test only related to starch, and it's just the idea of adding iodine. If it's black blue colour, then the starch is present. If not, it remains an orange-brown. So the next molecule we're going to look at is lipids. So your lipids, their main two types that come up: triglycerides. So they contain a glycerol backbone and 3 fatty acids, and phospholipids, which have your glycerol backbone and have 2 fatty acid chains and a phosphate group. So the fatty acids are hydrophobic, so they will repel water, and since triglycerides only have fatty acids, they will just repel water. So if you think like oil, that's all triglycerides, that floats in water and doesn't mix. Whereas the phosphate group is hydrophilic, it's attracted to water, so when you mix the phospholipids, they will form either a meso or a bilayer. We'll talk about that in a future live stream. So, they only partially repel water. So, when we talk about triglycerides, we've got 2 types, saturated and unsaturated. So this is all about the fatty acid chains. and they being saturated means that they don't have any carbon-carbon double bonds. So if you look at this chain here, it's a completely straight train, no double carbon-carbon double bonds. Whereas this one here has got a carbon-carbon double bond over here. That means it's unsaturated, and in this case, mono unsaturated. Mono being one. So yeah, this will change the properties of your fatty acid and certain triglyceride. So the more unsaturated it is, the more kinks in the fatty acid structure. So as you see, this one here is straight. This one here has a bend to it. So this bend will break up the structure of the fatty acid, meaning that they don't fit nicely together, and so don't form stronger bonds with each other, and so have like lower melting point. Yeah, so they are lower melting point, but yeah, they also will contain more energy in this carbon-carbon double bond. Phospholipids, yep, they only attract water on one side. You have 2 rows of phospholipids. I should probably add a diagram. That'll be done later. With fatty acids facing inwards, so they're in your middle bit, and then your phospholipids facing outwards on your out towards the water. This is so you can have water both sides of the membrane while having this lipid layer in the middle. These create up your bilayers, which make up the cell membranes and nearly all membranes found in biology. So our test lipids, we add your lipids to ethanol to your sample, and then you add water. Make sure you do it in that order. So ethanol first, then water. Then you're going to shake it up, and if a cloudy white emulsion forms, then lipids are present. If not, they aren't present. So proteins, this is our next structure. They are a polymer made up of amino acids. This is our general formula for an amino acid. So you've got this amine group here, the NH2. You've got this central carbon, and then you've got this acidic group here. Hence the name amino acids. This is a hydrogen, and this R group is your side group. This can be replaced with one of 20 different amino acids, and we'll talk about them again a bit more later as it comes up, both in this session and further down at other sessions. So the shape of protein is very, very important to its function. So to change the shape of a protein is to change its function, making it maybe non-functioning or improving its function. Yeah. So primary structure. Primary structure of protein is your chain of amino acids. These amino acids are bonded together by peptide bonds. That is between your amino group, your NH2, and your acidic group, your COOH. This is again a condensation reaction. So water is removed and you form your peptide bond shown here. So this again can be, this when it's formed, it requires use of ATP. That is wrong. Oh no, ATP itself is hydrolyzed. We'll talk about ATP next week. So, in the secondary structure, this is when our long chain starts to fold up. It folds up into 2 particular structures, your alpha helices and your beta pleated sheets. These structures are just held by simple hydrogen bonds. Next is your tertiary structure. This is when the length of one polypeptide one peptide chain, folds up into its shape. This shape sort of held by multiple different bonds, and then go on to the quaternary structure quickly. That is when you have multiple different proteins coming together to form your final protein. Think of hemoglobin. It has 4 proteins. Muscles, they have loads of different proteins. So the bonds that hold them together, they're found between your tertiary and quaternary, are your hydrogen bonds. So they are between like your OH group or NH groups. Ionic bonds, so anything that's like with a charge, or disulfide bridges, so anything with sulfur. So if you were to change the sequence of amino acids, so if you were to move cysteine, that contains a sulfur group. You don't have to know this, but as an example, that means you wouldn't be able to form a particular disulfide bridge, so the structure wouldn't be the same. And so then, going on to enzymes, you won't be able to have the same active site. So you can, let me think. So it won't be able to perform its function in the same way. So our test for proteins, this is quite a simple one. It's your Buret's reagent. As its sample, it goes from blue to purple pink. There is, from what I remember, there isn't too much in the difference in colour. It's just they're present. So enzymes. These are proteins and they have an active site. So your active site is the part which catalyzes the reaction. This is used we represent this with the induced fit model. So this is where the active site isn't the exact complementary shape to your substrate. So when they bind, the substrate will then form, the enzyme will then, active site will then form to the substrate. If we look here, shown there, so it's changing as it's going along. So it forms that sort of square from that circle shape. So let me think. So the enzyme substrate, so yeah, it becomes fully complementary, and then the substrate binds to it. This will then form an enzyme substrate complex. This puts stress on the bonds of the substrate. This lowers the activation energy needed to break the bonds in the substrate, allowing them to break at a much lower temperature than would be normal. So, how does temperature and pH affect enzyme action? Every enzyme has an optimum temperature for working and an optimum pH. So, temperature, if you were to think like one's internal enzymes are going to be like 37 degrees, there's their optimum. Whereas something like bacteria external enzymes, the ones they produce to like break down the environment, they are going to work at a much lower optimum temperature. So as temperature increases, you're going to see a nearly exponential growth of your activity until your optimum temperature is reached, and then it declines rapidly. This is due to bonds between the peptide, within the peptide, will have too much energy and break apart. This leads to the denaturing of the enzyme, so it will no longer function. Then you have pH. Optimal pH is generally quite a narrow range of a few pH. So if we look here at this one for one found in stomach acid, it's between 1 and 4. So anything beyond, even really between 2 and 3, so 1 and 4, it's denatured. So this is due to the enzyme wanting set conditions and not much beyond that. So if it's too acidic, then protons might interrupt it, or if it's too basic, it might lose protons. You don't have to know about that. Cool. So how does concentration affect enzyme action? So as the concentration of the enzyme of the substrate increases, so does the rate until the concentration of enzymes becomes the limiting factor. So once you have enough substrate, it will then saturate all the enzyme active sites. So all the active sites will be working, trying to break down the substrate, and this causes a plateau as you increase it until you increase the amount of enzymes, which will then increase the activity. This also works in reverse. So for the same enzyme concentration, so if you were to have the same substrate and increase enzyme concentration, it would keep increasing until you run out of substrate, and then you again reach that plateau. So enzyme inhibition, this is the last bit about enzymes. You've got two types, competitive inhibitors. These will bind to the active site of the enzyme instead of the substrate, forming your inhibitor enzyme complex. This will reduce the rate of reaction, but can be overcome by increasing the concentration of the substrate. Non-competitive inhibitors will bind to the allosteric site, basically another site found on the enzyme, which would then change the structure of the active site, change the shape of the active site. This will mean that it's no longer complementary to your substrate. This will then lead to the substrate not being able to bind, and so you'll have a near zero rate of reaction, and that cannot be overcome by an increase in the concentration of substrate. So question 1. You've got this figure here. Figure 2 shows a polypeptide. This is found on the cell surface with an NH2 group at one end and a COOH at the other. Describe how amino acids join to form polypeptides, and so that there's always an NH2 at one end and a COOH at the other. So this is going back to your amino acid structure. Because you form a condensation reaction between your COOH, so your acid group, and your NH2, your amino group, that means there's always going to be one left over on the one side, so the one that doesn't react. So on the side of the carboxylic reacting, the NH2 group's going to remain there, and on the one with the amino group reacting, the acid group's going to stay there. Let's have a look at the answers. So form that peptide bond, make sure you mention that, and so then you have the 3 groups on each side. So describe the biochemical tests you would use to confirm the presence of lipids, non-reducing sugars, and amylase. So start off with lipids. This is probably the easiest one. So we add ethanol to our sample, then water, give it a good shake, and we should form a cloudy white emulsion if lipids are present. For non-reducing sugars, we do your reducing sugars test, like I mentioned earlier. So you'll add Benedict's, heat it, should form a blue, shouldn't change, so no brick red precipitate should form. And then we have to get a new sample, boil it in acid, then neutralize that acid, and then So then do your Benedict test again, so heat it in the water bath. So the last one is amylase. So this is to catch people out, to understand that amylase is an enzyme that will break down starch. We'll talk about this when we talk about digestion and all that. So amylase, talk about, so one is the enzyme, so it's made up of proteins. So we have to test that it's made up of protein. So we do the burette test, and it should turn purple, pink. And then also, because it breaks down starch, we have to know, add starch to a solution, add amylase. If it turns, if the starch is still present after a set amount of time, 30 minutes or so, by doing the iodine test, then amylase isn't present. But if all the starch has been hydrolyzed by the amylase in that 30 minutes, then the iodine solution would stay that orangey brown, and so it isn't present. So it is present even. So going through that. Yep, Buret's test, starch, go on. ethanol, water, yeah. Benedict, yep. We've got this one here. This is talking about enzyme action. So washing powder contains enzymes. They've got it at two different things. So your activity at 30 degrees, it's here, 50 degrees here, 60 degrees here. So explain the shape of the curve difference. So at here, we are way beyond the optimum. So as time goes on, the high temperature breaks the bonds between the proteins, so your disulfide ionic hydrogen bonds. This then changes the structure of the active site, no longer forms enzyme substrate complexes, and so the reactivity decreases. Here, something similar is happening where some of the enzymes are starting to denature, but it's not all of it. So again, temperatures breaking the bonds, changing the active site, reducing its rate, maybe not fully denaturing it. So both denatured, fast denate, breaks hydrogen bonds, changes the shape of the active site. So describe how competitive inhibitors reduce the rate of reaction. So this is the idea of the inhibitor binding to your allosteric site or the another site on the enzyme, changing the shape of the enzyme, changing the shape of the active site of the enzyme, meaning it can't form your enzyme substrate complexes, so it can't act as your catalyst to your reaction. So here you've got pectin, found in some fruit and veg, acts as an inhibitor, I believe. The suggestion is that it's an inhibitor for your lipase enzyme. Use figure 1 to explain scientific results as sort of if you think it is or isn't a non-competitive inhibitor. So it is a competitive inhibitor because it has this near zero constant flat graph, even as your concentration increases. So if it was a competitive inhibitor, we would see a decrease in the rate, but it would still eventually reach the same rate as without it. But because the pectin's been added, it is that flat, constant, low level rate. Oh, yeah. So this is the end of this first stream. If you have any questions, let me know. This will be going on at the same time next week, so starting at 8:30, and we'll be going through topic 2 and topic 3. See you then.